And Neurona is a very interesting company, founded very early, 2008, founded by some UCSF very good neuroscientists. But the company's behavior is also interesting. If you go online, although they're a private company, even private company, usually you can at least find some copper deck. And I asked many people to try to find any copper slide deck from Neurona, nothing. But of course, they do have some on their website. They have posters, they have some presentation, and occasionally you can find a YouTube video that will give one talk. But in general, this company tried not to publicize, even do not want to share their copper deck. So that's also very interesting in terms of keeping some secret. But overall, a lot of information already public. And if people remember, in June 2024, I cover another company, Neurona Therapeutics, which they definitely focus on the focal epilepsy as one of the indication. So this space is also very interesting. There's unmet medical needs. And basically, I won't go into too much detail for BioSmart member. You can go back to the replay for June 2024 to understand more about epilepsy. It's a neurological disease, mainly happens at the cerebral cortex, or those hyperexcitability of those cortical neurons. It's basically a neuronal networks. People can pretty much use the EEG, that's the gold standard diagnosis. Every time you see, you're starting to see the epilepsy, I want to quickly introduce some terminology. One simple episode of kind of epilepsy is called seizure. Seizure only refer to one occurrence. But as the seizure become more chronic and recurrent, and then it become a disease called epilepsy, just a recurrent seizure. And there's about 1 in 20 people at least have one seizure in their lifetime, but only 1% of the whole human population can have this epilepsy, but still a lot. 1% is still quite a lot. There's different types of seizure. If people reading this Nature Review disease primer, there's focal seizure, meaning the area is relatively well-defined, but there are also some generalized all over the cortex. There's also some unknown kind of type of the epilepsy types. But right now, people mainly think the focal epilepsy represent the major patient population for a lot of drug development opportunities. And here is the rough numbers, because of course, Neurona only give the numbers for the US. Around the whole world, you may multiply by 3. So if US right now have about adult epilepsy patient, about 3 million diagnosed, and the whole world may be 9 to 10 million in terms of the number of patients. Among those, focal epilepsy represent the majority of those patients. About 2 million in US, let's say roughly multiplied by 3, around the world is about 6 million. Then among the focal epilepsy, one of the major unmet medical needs is the so-called drug refractory focal epilepsy, meaning you cannot just take any drugs because they're quite resistant. About 30% of the focal epilepsy patients are resistant to the drug treatment. That's why Neurona, when they were found in 2008, take quite a while to bring in either the embryonic stem cell derived interneuron or the iPSC-derived interneuron, try to see if there's any opportunity to bring the neuron cell replacement therapy into this space, because especially those drug-resistant subtype represent the major opportunity. And for patients other than drugs, they can also consider those non-destructive, but the destructive lobectomy, meaning doing some removal of certain area of the cerebral cortex so that we can reducing the hyperexcitability. So the hyperexcitability is mainly is an imbalance between the excitatory neuronal transmission versus inhibitory neuronal transmission. The inhibitory part is mainly mediated by the GABA neurotransmitter, which is a very famous inhibitory. In some way, you can even think, although it may not be exact, GABAergic neuron, you can think about as if they are the neural counterpart of Treg, regulatory T cell. It's the negative side of keeping the check and balance. That's why epilepsy is believed to have some deficiency in the inhibition, causing the hyperexcitability. That's why those companies try to see if we allogenically replace those interneurons and GABAergic inhibitory neurons into the cortex, there's an opportunity we may be able to reducing the epilepsy frequency. So this company is doing very good neuroscience, very solid scientific foundation, and they believe that epilepsy provide them the opportunity for the GABAergic interneurons. Of course, they started company with a lot of animal model, mainly the mouse model, try to dissect the lineage. If people reading the literature in the neuroscience or neurobiology, recent years with single cell RNA-seq become available, there's at least 50 types of interneurons in the cortex. That's just so many of them. You have to see the single cell gene expression pattern, and it's so complex. It's difficult to distill that in one webinar. But I just want to people point out that one of the area of the brain called MGE and the medial ganglionic eminence area, those type of neuron called the parallel GABAergic interneuron seems to be more relevant, deficient in the epilepsy patients. That's the main opportunity. And I also like to point out, refresh the point from June 2024. The main site of epilepsy in the brain is hippocampus. Those are the major sites. That's why you want to replace some of the interneuron. You want to inject the cells, neurons into hippocampus. That's the most people are doing right now. And I won't go into too much detail in terms of the company. It's a very good technology in terms of how to derive the embryonic stem cell and differentiate them into GABAergic interneuron. The really good news for people working with interneuron, cortical neurons, the time to differentiate into cortical neuron is pretty short, 2 weeks, 2 to 3 weeks. But if for people working on Parkinson's disease, takes several months, maybe can up to not just counting at least 1 or 2 months to get to the progenitor cell, but once you inject the progenitor cell into the brain, it takes at least about 5 to 6 months for them to become mature and grow to the neurite, the axon and dendrite. That's why differentiation into dopaminergic neuron is way more tedious and take longer time. Cortical neuron is definitely the low-hanging fruit. That's why I really like the company Neurona just picking the low-hanging fruit, because cortical interneuron is much easier to work with in terms of the manufacturing and differentiation. They build, because they are allogenic cell replacement company, they build those PSC cell bank. Allogenic company always need to build a multiple donor cell bank to prepare for those differentiations. Then they follow the developmental neurobiology very carefully by different, although they didn't never fully disclose the detail, you may be able to dig out from the pattern literature. They can patterning of the developmental biology and eventually getting those so-called parallel GABAergic interneurons. And most interestingly, they are not injecting progenitor. They can engraft post-mitotic neuron. That's the major difference from many of the neural cell replacement company. Many of the neuron types, you can only inject progenitors before they fully commit to some lineage. But for whatever reason, people not fully understood that this disease, epilepsy, they can succeed by injecting post-mitotic neuron. That's really very convenient, because post-mitotic, you don't need to worry about the proliferation. You can handle those neurons and in a pretty relatively straightforward by doing the cryopreservation. And then eventually you can even measure, because it's a mature neuron, you can do electrophysiology for the potency assay. For progenitors, as people in the field already know, Blue Rock, all those people know that measuring the potency for a progenitor neuronal types is very difficult because they're not neuron yet. But if you're dealing with post-mitotic neuron, you can do electrophysiology for the potency, which is a very convenient thing to have. Then, of course, the company spent so many years, they develop a lot of good preclinical data before they go clear the IND. They have a very good mouse model for one particular subtype, the temporal lobe epilepsy, MTLE. They show some efficacy in the rodents, the mouse model. I want to go into all the details. Basically, you focus on the hippocampus, and in the hippocampus, there's 3 major regions, CA1, CA2, and CA3. And not only is they important in the epilepsy pathogenesis, they're also important in learning and memory. That's why if you're just doing lobotomy treatment, sometimes that can have the side effect of hurting the learning and the memory. That's why doing a replacement therapy, you have the opportunity to really hopefully alleviate some of the side effects on learning and memory, cognitive side effects. And yeah, this is just in non-human primates, they did a very good job of finding a device to do stereotactic MRI-guided delivery of the interneuron into the cortical regions, mainly the hippocampus, which is a pretty very mature way of, because cortex compared to injecting to putamen, it's way more straightforward because it's relatively shallower in the brain. Then in terms of their phase 1/2 clinical trial design, they have basically an open label clinical trial without a treatment, without a sham surgery. Only later they will have stage 2. They haven't done the stage 2 yet, as to the best of my knowledge. In stage 2, they will introduce some sham surgery. Whether they call it stage 2 or phase 3, that's also remain to be determined. But so far, we only have the data for the stage 1 of the phase 1/2 trial. 5 patients in the low dose. The dose in cell therapy means the number of cells, but they haven't disclosed how many million cells they need to inject for the low dose and how many high dose of the cells they need to inject for the cohort 2. Each cohort have 5 patients each. They did a very impressive job opening 12 clinical sites in the United States, in many of the universities all over the United States. And the primary endpoint is just pretty straightforward. You just see the frequency of the adverse events, that's safety, because for cell replacement therapy, phase 1 trial, safety is always the #1 concern from FDA perspective. You don't want the patients to have any residual pluripotent cell, which can be a cancer risk. So that's safety. But they can also read out some pretty straightforward secondary endpoint for efficacy, which is just reduction in the seizure frequency. You can also measure the responder rate. And you can also put in some other endpoints, but definitely they have pretty interesting efficacy readout. Not only it's safe for the 10 patients so far, they have some interesting efficacy readout, because that you can see the seizure frequency for both the low dose and the high dose. And the low dose, you have 1 patient hasn't reducing much. I also want to point out, because it's allogenic cell replacement therapy, you need to have immunosuppression. Like Blue Rock for Parkinson's disease, they chose to have 1 year of immunosuppression. Then hopefully the brain is relatively immune privileged, so that after 1 year, when this engraftment settled, the immune system won't attacking those grafts. But that's remain to be an open question whether that's indeed the case. But people tend to believe after 1 year of immunosuppression, you may not need to go too much extensively. FDA will want people to follow up whether that hypothesis is true. But you can see the low dose, and about 4 patients respond very well. 1 patient didn't respond. Then in the high dose, all 5 patients respond, having a significant reduction of the seizure frequency. So that data looks pretty straightforward. That's why in 2024, Neurona was able to get the RMAT designation. RMAT stands for Regenerative Medicine Advanced Therapy designation. It's the equivalent of breakthrough therapy in antibody and small molecule drug space. So it's a very good thing to have. That's why they can, based on this RMAT designation, they can talk with FDA and expect to launch the phase 3 trial. They may no longer wait for the stage 2 of the phase 1/2 trial and just go straight into a phase 3, what they call an EPIC clinical trial. And based on the RMAT designation in June 2024, it's a very promising company. I think they're going to do IPO maybe when the climate is doing better in terms of 2025 and 2026. But overall, I think the company's science is very solid, and the efficacy data looks pretty impressive. So that's all I have to share, and invite people to ask questions from the audience if you have